Good morning. So the significance and translation readiness of our nanotherapeutics efforts in my laboratory at UCLA addresses two important areas. Number one is the custom design of nanocarriers to improve chemotherapy delivery for cancer, including delivery of chemotherapeutic agents that can also wake up the immune system and synergize with checkpoint blocking antibodies. The second major area is the use of a nanotherapeutic platform for treatment for autoimmune and allergic disease processes, which carry an even bigger economic burden than is the case for cancer. I am a professor of medicine at UCLA and founder of the Division of Nanomedicine at UCLA. I also serve as the research director for the California Nanosystems Institute, where I oversee some of the team science efforts driving translational research, some of which end up in our Magnify incubator in CNSI. My research has been well funded by the National Cancer Institute and the NSF, as well as innovation fund awards from the UCLA School of Medicine. I will briefly tell you about two of our major research efforts that have been translated into startup companies. The first startup company is Westwood Biosciences, which have been issued 2 US patents to develop the silicosome platform for the treatment of pancreas and colon cancer. The silicosome platform is the equivalent of a liposome, except that the lipid bilayer covers a solid hollow space that has a lot of drug delivery capacity, allowing it to outperform a commercial liposome Onivide in preclinical treatment of pancreas and colon cancer. The platform is currently in a preclinical stage, having achieved upscale GLP manufacturing and is awaiting IND filing. We have recently demonstrated that irinotecan also has immunogenic properties that allows this platform to be synergistic with checkpoint blocking antibodies. The second startup also expands on the idea then of immunogenic cell death therapy by delivering dual delivery liposomes. One drug, usually a chemo agent, induces immunogenic cell death, and the second drug is interference in immune escape pathway, leading to propagation of the immune response. A US patent has been filed and licensed to nanotherapeutics, which is in the preclinical development stage for immunotherapeutics intervention in heterogeneous immune landscapes, including triple-negative breast cancer. A new platform that I want to pitch for partnerships and research collaboration aims to harvest the special ability of the liver to suppress immune responses elsewhere in the body by inducing a unique class of suppressor T cells, also known as regulatory T cells, or for short Tregs. It's been known for a long time that the liver is an immune privileged organ that has the capability of inducing immune tolerance to foreign substances such as food allergens or autoimmune disease antigens, and can be used as the target for tolerogenic immunotherapy. In this platform, we produce a nanoparticle that is comprised of an FDA-approved polymer to target the liver after intravenous injection. The particles home in on the liver sinusoidal endothelial cells, which also function as the antigen-presenting cells to generate the regulatory T cells. If we include food allergens or autoimmune antigens in the tolerogenic nanoparticles, it is possible to suppress food-induced anaphylaxis or a diabetic state, as demonstrated by the collaborations in the Innovation Fund Awards from the School of Medicine. I outlined for you a recent UCLA newsroom release that highlights the use of our platform for the treatment of anaphylaxis against an egg allergen. We demonstrate that allergen delivery to the liver by the tolerogenic nanoparticles could prevent anaphylaxis, both as a preventative as well as a therapeutic treatment. The success could be duplicated by only using a tiny fragment of the egg allergen to achieve the response outcome. Success was also recently achieved by developing a small fragment from one peanut allergen that could totally suppress the anaphylactic response induced in a model using crude peanut allergy extract. The intellectual property generated has resulted in the filing of a comprehensive UCLA patent, pitching the use of polymeric nanoparticles that target liver sinusoidal endothelial cells to induce antigen-specific tolerance. It has been supplemented also by constructing that particle to also deliver a tolerance to viral vectors that is used for gene therapy. The disease areas that are covered by the patent include its use for food allergies, inhalable allergies such as for asthma, anaphylaxis, including to therapeutic substances, treatment of systemic and organ-specific autoimmune disease such as type 1 diabetes and lupus, life-threatening allergic drug allergies, and then immune responses to viral vector therapeutic delivery. We look forward to discuss and develop this platform with commercial partners in one or more of the disease applications shown on the left-hand side, and we are also open to research collaboration on these treatment goals. Thank you.